Thanks for coming to our distinguished lecture today. I'm Marcus Baumann. It's good to see all of you. This, as you know, is a monthly lecture series. And once a year, we honor one of our own, Dr. Gary Hunter, who's with us today. Just to give you a little bit of a backstory, in 2013, actually in 2012, many of us started hearing rumblings that Dr. Hunter may be on the verge of retirement. None of that was directly from him, but that became a rumor. And so we thought very quickly, I mean, essentially Dr. Hunter, as all of you know, is the godfather of exercise biology at UAB. We thought we've got to do something before he escapes us. So we created and defined an annual Gary R. Hunter Distinguished Lecture as part of our series. We all should have known better that in 2013 and 14 and so forth, Dr. Hunter would continue to submit grant applications and continue to publish, and he wasn't going anywhere. So it's great to have this opportunity in our fourth year of giving out the award to have him with us. And afterwards, we'll take some pictures. I want to make sure you hang around for that. I know you love pictures. But what we've done every year is try to bring in a prominent scientist that has made seminal contributions to the field, someone who we think has like-minded both processes and sort of a career path similar to what Gary has accomplished. And we've brought in, as you know, some accomplished scientists over the years, and this year is absolutely a perfect example of that. Very, very happy to have Dr. Doug Seals with us from the University of Colorado at Boulder. Give you a little bit of background. Dr. Seals has a pretty amazing training history, a pretty amazing pedigree in the field. As a trainee at University of Wisconsin, he actually overlapped with Gary by about a year. Gary was a young assistant professor, and Dr. Seals was a trainee at that time. He then left there, did his postdoc at probably the premier laboratory in the world, John Holloway's lab at the University of, Washington, St. Louis. If you know the lineage of people who have come out of Holloway's laboratory, it's pretty incredible. Dr. Seals being one of those individuals. He then went to train with Charlie Tipton, University of Arizona, where he was on faculty for several years. Another legend in the field. And then made his way to University of Colorado Boulder. And over that span of time, he's published over 300 papers, high-impact papers, focused primarily on the cardiovascular system and aging biology, and he's here to tell us a little bit about some of that work today. So it's a great honor to have Dr. Seals with us. It's a doubly great honor for me to be able to present you with this year's Hunter Award. We'll get a few pictures after the lecture, but welcome. Thanks.
Thanks, Marcus. Thanks, Marcus. And it's a great privilege to be here and participate in this lecture series, and of course to be able to honor Gary. So I'm very grateful for the consideration that led to the invitation today. Thanks, Marcus. So I'd like to talk with you about something that you may not hear much about. It's certainly not a common discussion topic, but vascular aging is something I want to talk with you about today, and the role of aerobic exercise in facilitating healthy vascular aging. So this is the outline for what I'd like to cover in the next 45 minutes. Start with some background and significance just to set the context, and then talk about the influence of aerobic exercise on two primary processes that go on in your arteries as you age. One is related to large elastic artery stiffness, and the other to the development of endothelial function or dysfunction, and I'll talk more about those in a moment. And then wrap up with a short section on some newer data suggesting that aerobic exercise may actually exert direct protective effects on aging arteries. Let's talk a little bit about the context of this work and why I think it's important. Cardiovascular disease prevalence has actually been declining for the last few decades, but these set of diseases remain the leading cause of morbidity and mortality in the U.S. and other industrialized countries. And this high prevalence of cardiovascular disease is a direct relation to advancing age. So Cardiovascular diseases, like most chronic diseases, are diseases of aging, and aging is the primary risk factor for cardiovascular diseases. And indeed, most cardiovascular disease occurs in middle-aged and older adults. Now, this is going to become a really even greater problem in the future because of the interaction between aging and cardiovascular disease and simply the number of older adults that are in the pipeline. This is a American Heart Association policy statement from a few years ago forecasting the future of cardiovascular diseases. And I'll just highlight 3 points that are relevant to the discussion today. One is, in less than 15 years, 40% of US adults will have at least one form of cardiovascular disease. Secondly, importantly for our discussion today, these changes will be driven primarily by the aging of the population, and also important for our discussion related to exercise today, effective prevention strategies will be needed if we're going to do anything about this. Well, of course, it would be very helpful for us to know exactly how aging increases our risk of cardiovascular disease if we're going to figure out how to intervene and either reduce or prevent this. And we now know that most of the cardiovascular disease that develops with advancing age is actually due to this intermediary step by which arteries develop some type of dysfunction. In fact, not only is this age-associated arterial dysfunction a key player in the development of cardiovascular diseases, but we now know, based on epidemiological data, that it's linked to a variety of other common chronic disorders of advancing age, many that you'll recognize here. Some of you work in these areas. So this is really something that we want to try to delay or minimize or even prevent if we can with advancing age. Now, several kinds of changes occur with aging to arteries that contribute to increased risk of these chronic disorders, but two that we think are very important and which we've discussed, that we've investigated in our lab, are increases in arterial stiffness and the development of endothelial dysfunction. Let's talk a little first about this increase in arterial stiffness. Now, the stiffening of the arteries that occur as you age is confined to what are called the large elastic arteries, and these are the aorta and the carotid arteries. These arteries, as the name implies, are designed to accept the blood that's pumped out of the heart with each contraction, expanding to accept that blood, and then using their elastic properties recoil to create the kinetic energy necessary to drive blood flow down to your tissues during the relaxation phase of the heart. Now, as you might imagine, when we're young and healthy, the ability to expand and recoil is quite robust. The arteries are highly elastic. But as we age, these arteries begin to stiffen, they lose their elasticity, their ability to expand and recoil. And this leads to a whole host of very serious pathophysiological problems. One, it increases our systolic blood pressure, which is the blood pressure that increases as we age and can lead to hypertension. And secondly, it increases what we call the pulsatility of blood pressure and blood flow. And this increased pulsatility is actually transmitted to our tissues and is particularly damaging to what they call high flow vital organs, which include the brain, the heart, and the kidneys. The endothelial dysfunction that develops with advancing age refers to dysfunction of this endothelial lining of your arteries, this single cell layer at the interface between where blood flows and the walls of the arteries. And these cells, these endothelial cells, are like small endocrine organs. They produce a wide array of biologically active molecules that act in autocrine or paracrine fashion to influence the function and the health of the arteries as well as the surrounding tissues. Probably the most important molecule produced by these endothelial cells is nitric oxide, the gaseous signaling molecule, and that's protective to arteries. So impaired endothelial function with aging and reduced nitric oxide bioavailability are key antecedents to the development of atherosclerotic diseases, including coronary artery disease. Now, the mechanisms by which aging leads to vascular dysfunction with age are still under investigation, but we know that two major processes are important. One is the development of oxidative stress within your arteries as you age, and this is driven by excessive superoxide production, a very important reactive oxygen species in arteries. And the second is the development of this chronic low-grade inflammation that features elevated levels of pro-inflammatory cytokines. And these processes are highly interactive. They amplify each other and eventually create this sort of vicious cycle where they feed on each other. And this produces a lot of downstream effects. One is that it reduces nitric oxide bioavailability. But this process also actually affects the arterial wall, and in particular the extracellular matrix. And there are three main changes that occur in the extracellular matrix of your arteries as you age that contribute to particularly arterial stiffening. One is collagen, which is the main structural protein conferring stiffness to arteries, increases its expression. So collagen is deposited, quote unquote, in your arteries at a higher rate. It's a sort of a pro-fibrotic process that we see in many diseases, but perhaps at a lower level. Now, elastin is a structural protein that confers, as the name implies, elasticity to your arteries. And elastin fibers become fragmented and disarrayed with advancing age. This also contributes to the stiffening of your arteries. And the third change to the extracellular matrix is the development of what are called advanced glycation end products, or AGEs. And these are molecules that cross-link these structural proteins. And that also incurs additional stiffness to arteries with advancing age. So these are the changes that occur in the extracellular matrix. We've been able over the last couple of decades to develop a pretty unique translational research approach to studying vascular aging. So given a particular question or intervention that we want to study, we may study it first in cells or mice or people. We can translate forward and we can reverse translate. I'll talk a little bit about that today. Our ability to study people was really aided in the late 90s when I was able to establish an NIH clinical research center at the University of Colorado, Boulder, which is an undergraduate campus. So it's the only clinical research center supported by the NIH that is actually on an undergrad campus. Every other clinical research center supported by that program is at a medical center or a hospital. So this really helped us to translate these observations to people. I'd like to spend the rest of the time talking about the effects of regular aerobic exercise on these processes that I've just introduced. And let's start with large elastic artery stiffness and how exercise may influence that with aging and also the mechanisms by which it might do that. So we've studied aerobic exercise in the context of both preventing age-related increases in arterial stiffness and also more as a treatment in middle-aged and older adults who already have some level of stiff arteries. So I'll present data in both of those contexts. Aortic stiffness, or the stiffness of the aorta, is assessed with a measurement called aortic pulse wave velocity. You may or may not have heard about this measurement. But essentially what this involves, and as the name implies, is we're measuring the velocity at which a pulse travels down a segment of the aorta after blood is ejected out into the aorta from the heart with a cardiac contraction. The faster the velocity, the stiffer the artery is the interpretation scheme. Using this technique, this is actually our first study ever on this topic, we measured aortic pulse wave velocity in groups of healthy women. And the initial observations were that among healthy non-exercising women, the postmenopausal women had much higher levels of aortic stiffness as determined by aortic pulse wave velocity compared with premenopausal controls. But when we brought in endurance exercise trained women into the laboratory to perform the same studies, we didn't see these significant group differences, suggesting that regular aerobic exercise somehow inhibits or tends to prevent the development of stiffening of the aorta. And since this early study, many studies have come out from other laboratories confirming this, in both men and women. So it's well established now in the literature. We can also look at the effects of exercise and the stiffening of the carotid arteries. Here we take a slightly different approach. We use ultrasound imaging of one carotid artery to measure dynamic changes in the internal diameter of the carotid arteries during cardiac cycles. And on the contralateral carotid, you use something called a tonometer to measure excursions in blood pressure at the same time. And you can use those measures to actually calculate compliance. Now, compliance is the inverse of stiffness. So I'm going to show you data next in which carotid artery compliance is declining with age. So the carotid arteries are stiffening, but compliance is decreasing. So it's a little confusing. So here are data from some initial studies that we performed. The data in the men are shown in blue, and the data in women are shown in gold. And this is carotid artery compliance. You can see among late middle age and older healthy adults, it's significantly lower than what we see in young adults. And this is in the absence of any kind of chronic disease. But when we look at middle age and older adults who regularly perform aerobic exercise, carotid artery compliance is much better preserved than what we see in their sedentary peers. So this also suggests this prevention of development of stiffness in these particular arteries with aging. We can then take these groups of middle-aged and older adults who don't exercise, and we can ask the question, is exercise a treatment or medicine for this particular malady of arterial aging? And this is the aerobic exercise training program that we've used through the years. People exercise on their own. The mode is brisk walking for about 40 to 45 minutes a day, most days of the week, for about 3 months, and at a moderate exercise intensity. So all the training studies that I'll refer to as we go forward will be using this intervention. And with this intervention, what we see in both middle-aged and older men and women is about a 30% improvement in carotid artery compliance after only 8 to 12 weeks of this brisk walking program. So that's quite a robust finding, and it does indeed suggest that aerobic exercise can be viewed as a very effective medicine for age-associated arterial stiffening. And we've tried to look at the mechanisms by which aerobic exercise confers these benefits on arterial stiffness with age. And the problem is that these arteries are in a difficult location to actually experimentally manipulate. And so we've had to go to our mouse model and take a reverse translational approach to this. And what we've done is used wheel running as a model of voluntary aerobic exercise in people, in these mice. And we've used about a 12-week period of this wheel running exercise, and we've started it later in life. So this is analogous to starting an aerobic exercise intervention in late middle age and older people. So it's very similar, and that was by design. We can measure aortic pulse wave velocity in these mice under light anesthesia, so we have a translational measurement that we use in people and in mice that is exactly the same measurement. And here in the lower corner just is intended to remind me to tell you that we see the same effects of aging and exercise in the mice that we see in people. So higher aortic pulse wave velocity in old mice who don't exercise compared with young mice. And then 12 weeks of exercise reduces the aortic pulse wave velocity, significantly lowering it to levels that are not different from young mice. So very similar to the data in humans. We've sort of validated the model. We can now take the arteries and we can perform various biochemical assessments to look at some of these mechanisms that we talked about. We've used a marker called nitrotyrosine, and I'll talk a little bit more about that later, as a marker of oxidative stress in these arteries. And so if you look at the aortas of these three groups of mice and you look at nitrotyrosine abundance in the arteries, this is the aorta from the different groups, you'll see that the aorta of the older mice who don't exercise have much higher levels of staining of this marker of oxidative stress than the young controls. 12 weeks of wheel running exercise completely reverses this profile. So it ameliorates this marker of oxidative stress that we see with advancing age, suggesting this could be a mechanism involved. Similar results when we look at markers of inflammation. So these are 3 of the many pro-inflammatory cytokine-based markers that we've assessed of inflammation. And what we see is the same pattern. In older mice who don't exercise, their aorta has high expressions of these pro-inflammatory cytokines compared with young controls. And 12 weeks of wheel running exercise reverses this profile, reduces the expression of these pro-inflammatory cytokines that we see in the aortas of young animals. So there's a very potent anti-inflammatory effect of aerobic exercise in this setting. This also allows us to go in and look at mechanisms related to these changes in extracellular matrix that we just talked about. And what we find is that we don't observe any influence of exercise on this fragmentation of elastin molecules that I talked about, where the elastic fibers fragment with age. We don't see any effect of exercise on that, but we do see effects on both collagen deposition in the arteries and also the abundance of these advanced glycation end products, these chemical crosslinks. These are data from collagen 1, which is the primary load-bearing structural protein in arteries. And this is from carotid arteries. So here we're using immunohistochemistry to look at expression of this structural protein. And you can see in old mice who don't exercise, they have much higher levels of this stiffening-related structural protein, collagen 1. And this is the whole vessel in blue compared with the young controls. It's primarily due to depositing collagen in the outer or adventitial layer. That's sort of a sub-story we won't get into today. But the most important thing is after 12 weeks of wheel running, we completely reverse this sort of low-grade fibrosis process. So total collagen is reduced, and there's a suppression of collagen in both the outer adventitial layer and also the medial layer, the middle layer of the artery. So exercise exerts this anti-fibrotic effect on aging arteries. And finally, we've looked at these advanced glycation end products that confer additional stiffening to arteries with advancing age. And we find that those molecules are elevated, they're more abundant in the arteries of old mice who don't exercise compared with young controls. Again, about 3 months of wheel running exercise markedly suppresses the abundance of these chemical crosslinks that we find in the arteries. So this is just a quick summary of this first section, or this last section of the talk. Aerobic exercise has a very potent effect in preventing and reversing arterial stiffening with advancing age. That's associated with a suppression of the increase in oxidative stress and inflammation that typically is associated with advancing age. And we see downstream effects in reducing the amount of collagen deposited in arteries and also the number of these advanced glycation end products that are formed. Okay, let's talk a little bit about exercise and endothelial function and whether this can prevent endothelial dysfunction with advancing age. We've looked at this, again, from a standpoint of can exercise prevent the development of endothelial dysfunction, and in people that already have it or animals that already have it, can we reverse and restore endothelial function through exercise? Well, how do we measure endothelial dysfunction of this single cell lining of our arteries? Well, the bioassay for that is really reduced vasodilation. That's the way we measure it. And this reduced vasodilation is in response to either a chemical stimulus, and that's usually acetylcholine. So if you read the literature, that'll be one way that we stimulate this dilation. And the other is a mechanical stimulus that is most often an increase in blood flow or shear rate. Either of these induce a nitric oxide associated or dependent vasodilation. And so this is called an endothelium dependent dilation. So if you see EDD, that's what that's referring to. Endothelium dependent dilation that's nitric oxide dependent. And arteries from old people and older mice consistently show this impaired endothelium dependent dilation, which again, try to keep in mind, is simply a measure of endothelial function in health. Okay? So this is the setup for chemical stimulation of this dilation in people. It's a forearm blood flow model where we infuse acetylcholine into the brachial artery. This is done in a clinical research center, obviously. And then we have a strain gauge on the forearm that sends a signal as blood flow is increased in response to this dilating substance, acetylcholine. Now, these are data from our initial study back in the late 90s that we published in 2000, and we're looking at peak forearm blood flow in response to this acetylcholine stimulus. And in gray here are older men who are completely healthy but don't exercise. And you can see they have about half the endothelial function that we see in young adults. In men who are essentially masters athletes, endurance athletes, they have completely preserved endothelial function. Same age as these men, but they exercise, and these folks don't. We can then again take a subgroup of these men here that don't exercise, older men, and have them undergo that exercise training program that I just spoke about. And we completely restore their endothelial function. Now, these effects of exercise appear to be due to the ability of exercise to increase nitric oxide bioavailability, because when we repeat these experiments after blocking nitric oxide production with a chemical, we abolish these group differences that are due to exercise. So exercise training increases nitric oxide bioavailability, which improves endothelial function. That's the sequence of events. You can show the same thing when we stimulate a dilation with blood flow. And I show this in part because this is the most common way that we assess endothelial function in people because it's non-invasive. It's called brachial artery flow-mediated dilation. And it's a blood flow stimulation, again, of endothelium-dependent dilation. Here we're taking an ultrasound machine and we're imaging the brachial artery just above the crease in your elbow up here. And we can establish the baseline diameter of the artery. So that's shown in the red bracket here. This is the internal diameter of the artery where the blood's flowing. So that's how much it's dilated at baseline. And then we can pump a blood pressure cuff up on the forearm and leave that at a high level of pressure, including blood flow to the forearm for a few minutes. Doesn't hurt the forearm. And then we can release the cuff. And when we do that, there's a surge of blood through the brachial artery to try to perfuse that temporarily ischemic forearm circulation. And that stimulates mechanoreceptors in the endothelial lining, produces nitric oxide, and induces a dilation that we can measure with the ultrasound images. So you go from here to here. So the greater the dilation, the greater the endothelial function and health. That's the bottom line. So using this technique, we see very similar responses to brachial artery flow-mediated dilation. This is a group of older non-exercising men, completely healthy, have about half of the flow-mediated dilation that we see in younger men. Older men who exercise regularly completely preserve function. We can take a group of these older non-exercising men, have them undergo that exercise program. This was an 8-week brisk walking program, and we can increase endothelial function by about 50%. And if you look at the levels that they reach, it's really not that different from what we see in chronic aerobic exercising men, older men, and also in young adults. Now you'll notice in the last couple slides I've been talking about data in men. And that's because one very interesting sidelight to this story is that exercise does not produce the same improvement in endothelial function in postmenopausal women who are estrogen deficient. Very interesting. However, it gets more interesting because when we restore circulating estrogen levels by administering estradiol to postmenopausal women, bring their estrogen levels back up to premenopausal levels, and then have them undergo the aerobic exercise training program, we restore this ability to exercise to improve endothelial function. So estrogen appears to play a permissive role in transducing this exercise stimulus to the endothelial lining and function of the endothelium with aging in women. We've investigated the mechanisms by which aerobic exercise has these beneficial effects. We've focused on oxidative stress and inflammation largely, and I'll just emphasize those data in a few minutes here, to see how exercise reverses or prevents this endothelial dysfunction. These are some of the key events that occur that you can sort of try to experimentally isolate in people, and we've also done these studies in mice, but I'll focus on our studies in humans here. We've talked about the important role of superoxide and excessive superoxide production in creating an environment of oxidative stress in aging arteries. This, of course, is opposed by antioxidant enzymes, and the primary family of enzymes are the superoxide dismutases that neutralize the superoxide, keep that under control. We know that superoxide readily reacts with nitric oxide to form a highly reactive species, peroxynitrite, which turns out to be the species that actually causes a lot of the nitration of tyrosine residues on proteins that gives us this marker of oxidative stress that we mentioned earlier, nitrotyrosine. And finally, we've looked at the interaction between oxidative stress and inflammation by assessing various pro-inflammatory cytokines, and also NF-kappa B, the master pro-inflammatory transcription factor. So let me just walk you through a couple of the experiments where we've tried to piece this apart in people with advancing age and the effects of exercise. So first, what's the role of excessive superoxide production in all this? On the left are the data that I just showed you: brachial artery flow media dilation, lower with sedentary aging compared with young controls, well preserved with exercise with advancing age. If we now ask the question, what's the role of excessive superoxide? To get at that, we can bring these people into our clinical research center, and we can infuse very high concentrations of the antioxidant vitamin C. Much higher concentrations than you could ever get taking the vitamin orally, so don't get confused. This is not an intervention study. This is using vitamin C in super physiological concentrations to reduce temporarily superoxide bioavailability in the arteries and see what role that's playing in producing this arterial dysfunction. And when we do that, we completely restore, within a half an hour, we completely restore endothelial function in these older non-exercising adults. So that suggests that their chronically impaired endothelial function is indeed the result of excessive superoxide production. No effect on older adults who exercise. And that suggests that their preserved function under normal conditions is due to an absence of excessive superoxide-associated oxidative stress. That's the interpretation, at least. We can then look at, for direct molecular evidence of exercise on this marker of oxidative stress, nitrotyrosine. And we do this by utilizing our brachial artery catheters, and we're able to use fine J wires, and this is all done in a clinical research center, to go in and sample endothelial cells from our human volunteers, and we can take those cells and we can treat them and use quantitative immunofluorescent analyses to look at various protein markers of mechanisms of interest. So in this case, we looked at nitrotyrosine, again, our marker of oxidative stress, in these endothelial cells from three groups: the young adults, older non-exercising adults, and older exercising adults. And in the endothelial cells, you see high levels of staining of nitrotyrosine in the cells from older adults who don't exercise, suggesting direct molecular evidence that they have oxidative stress in their vascular endothelium. That's really a fair interpretation. But in the older exercising adults, their endothelial cells don't show this oxidative stress with advancing age. It's really remarkable. We've also looked at the possible role of upregulation of this enzyme that counteracts superoxide dismutase, and asked the question, are these levels higher in older adults who exercise? That might be a way where they that might mediate their lower oxidative stress. And so we used the technique to look at manganese SOD, the mitochondrial isoform of superoxide dismutase, in these same groups. Found that this antioxidant was lower, expressed in a lower level in the endothelial cells of older adults who don't exercise compared with young controls, well preserved in the endothelial cells of older adults who exercise. And then we were able to also look at extracellular superoxide dismutase. This superoxide dismutase is actually attached to the endothelial lining, the luminal side of the endothelium, and you can inject heparin, and that will break these bonds and free up the extracellular sod so that you can actually capture it in blood samples, and then you can assay for extracellular sod activity in these human subjects. And we see the same exact pattern where the extracellular sod activity is lower in older adults who don't exercise compared with young controls, well preserved in older adults who regularly perform aerobic exercise. So enhanced antioxidant enzyme activity may play a role in the lower oxidative stress with exercise. We've also been able to look at markers of inflammation. And here we're on the left looking at a pro-inflammatory cytokine, interleukin-6. And what we see is in the older sedentary adults, they're in their endothelial cells, much higher staining of this pro-inflammatory cytokine, but not in the endothelial cells of the older adults who exercise. We were interested in the possible role of NF-kappa B as this master pro-inflammatory transcription factor. We see the same pattern with NF-kappa B in the whole endothelial cells, higher with sedentary aging, well preserved, lower with exercise with aging. And we wanted to look more at the causal association of this elevated NF-kappa B mediated pro-inflammatory signaling. So to do that, we actually performed functional experiments to support this link. And here's just measurements of brachial artery flow mediated dilation, that measure of endothelial function. And under control conditions, placebo conditions, it's higher in older exercising adults than in their sedentary peers. We talked about that, no surprise. Then we treated both of these groups with an anti-inflammatory drug that inhibits NF-kappa B signaling, salsalate, that's been used in chronic inflammatory disorders clinically. Treated both these groups for 4 days. We found that 4 days of anti-inflammatory treatment completely restored function in the older non-exercising adults, no effect on the older exercising adults. And so the interpretation here is that the chronically impaired endothelial function of the older non-exercising adults is influenced by inflammation, because when you take the inflammation away, you restore function. And the exercising older adults do not have that inflammatory burden in their arteries as they age. And we performed vitamin C infusion experiments to go along with these to show that this was linked to oxidative stress, as we talked about earlier. So this is just a summary of this second section of data, at least, that regular aerobic exercise tends to preserve endothelial function with advancing age. It can be viewed very much as a medicine, a very effective medicine for restoring endothelial function with advancing age. And it does so by enhancing nitric oxide bioavailability, increasing the expression of superoxide dismutase in the endothelium, lowering oxidative stress, reducing superoxide, and also suppressing the development of inflammation in your arteries as you age. Some of the most exciting work, I think, on exercise in the setting that we're studying is recent data suggesting that aerobic exercise exerts a direct protective effect on aging arteries. And this is essentially suggesting that it shields our arteries as we age against a variety of adverse factors that our arteries are exposed to throughout a lifetime. For example, we've studied in humans the influence of 2 conventional risk factors for cardiometabolic disease: elevated LDL cholesterol and impaired fasting blood glucose levels. And if you look at a measure of endothelial function like brachial artery fovea dilation, you see a reduction in function in older adults who don't have any risk factors for cardiometabolic diseases. So this is an effect of aging. However, if you add either one of these risk factors, you see a further reduction in function. So it's like a double hit or a double whammy. So this is aging plus the presence of at least one of these risk factors. However, if you study older adults with these risk factors but who exercise regularly, they have completely preserved endothelial function. So it's as if the exercise is protecting these arteries from both age and these risk factors, well-established risk factors for cardiometabolic disease. We wanted to gain more insight into the mechanisms by which this occurs. So we went to our mouse model and we used as a measure of, or a stimulus of an adverse factor, Western diet. And we designed a diet to give to mice that is very similar to the Western diet that many people in the United States and other modern societies eat. High fat, high sucrose diet, and in about the same percentages. And we particularly wanted to know the influence of oxidative stress in transducing a deleterious effect of Western diet on aging arteries, and importantly, how aerobic exercise might prevent this, given the studies that I just showed you in humans. So here's the experiment. We performed measures of endothelium-dependent dilation on isolated carotid arteries from mice. So here's the endothelium-dependent dilation. Remember, that's a measure of endothelial function. And in various groups of mice. So this first group are young controls. They have very robust endothelial function. If we feed mice a regular, healthy mouse chow diet, low fat, low sugar diet, and we follow them throughout age, older mice will demonstrate a significant reduction in endothelial function compared with young mice. No real surprise there. Now, if you take mice and either feed them just for about 3 months in old age or throughout a lifetime with a Western diet instead of that healthy mouse diet, what you see is a further decrease in function. So this is the double hit or the double whammy, right? This is due to age plus eating a Western diet. And so this is really representative of most people in the country probably. We wanted to know if this decrease in function with aging and Western diet was due to excessive superoxide bioavailability and oxidative stress. So we incubated the arteries with a superoxide dismutase mimetic, a mimetic of the endogenous antioxidant enzyme, which eliminated superoxide or significantly reduced it, and we completely restored function. So that suggests indeed that this decrease with aging and Western diet is excessive superoxide mediated. However, when we looked at the effects of aging and a Western diet on endothelial function in mice, we allowed mice access to voluntary wheel running for 12 weeks late in life. They had completely preserved endothelial function. So again, exercise appears to protect these arteries, and Temple had no influence on this response. So the older mice that had access to running wheels didn't have oxidative stress, at least at this level. Now this suggests that there's too much superoxide being produced and getting to the vascular endothelium, but we have no idea where that excessive superoxide is coming from. So last set of experiments I'll show you real quickly here. We looked at mitochondrial superoxide production. So as cells age, including artery cells, mitochondria develop some relative degree of dysfunction. And one of the phenotypic changes that occurs is they produce more superoxide in their electron transport chain. And so we wanted to look and determine whether this was a possible source of that excessive superoxide that we measured. So what we see is mitochondrial superoxide produced in the aorta now, so this is from arteries, is increased in old mice who eat a healthy diet, is further increased in the aorta of old mice who eat a Western diet, but in old mice who eat a Western diet but have access to running wheels, it's lower. So it's sort of the inverse of what we see with function, suggesting a possible role. So we performed more experiments looking at this mitochondrial dysfunction and its role in explaining this influence of exercise on Western diet. And this is what we found. So this is the last major data slide I'll show you. So thanks for being so patient. Here's the endothelium-dependent dilation, measure of endothelial function. And this is in 3 groups of mice, but all fed a normal chow diet. So the pattern is something you're used to seeing now. Reduced function with aging in the absence of exercise, and then well-preserved endothelial function in old mice who have access to running wheels. Now, we then took these 2 groups and treated the arteries under various conditions. In this first condition, we gave a chemical to the bath that incubates these vessels that has a chemical rotenone, which at low levels stimulates the production of superoxide from mitochondria in those arteries. And what you see in the old arteries from mice who don't exercise, they have a further impairment in function. So once again, a double whammy. Here's the effect of age. Here's the effect of age and this additional mitochondrial oxidative stress that we chemically induce. Here's the response of the old mice who exercise. They're completely impervious. They're resistant to this combination of age and this mitochondrial oxidative stressor. We can now try to recapitulate our in vivo Western diet data by incubating the vessels essentially ex vivo in a simulated Western diet, high palmitate and high glucose in this case. And the arteries from the old mice take another double hit. So they go from here in the red to this level of dysfunction. The arteries from the exercising mice, they're resistant to this ex vivo Western diet. And the question was whether these differentiated responses were due to mitochondrial oxidative stress. So the last set of experiments we performed is we repeated these experiments but added a mitochondrial specific antioxidant called MitoQ. And this should reduce or eliminate the superoxide being produced by mitochondria. What we found was we completely restored function in the old mice who had impaired function here to levels here. So that suggested that this impairment was due to excessive mitochondrial oxidative stress. But the arteries from the old exercising mice were not affected by MitoQ, suggesting that they did not have elevated mitochondrial superoxide production. And that's what led into this resistant profile. And when we look at markers of mitochondrial health, and I won't even go through these data, but these were all different markers, protein markers of mitochondrial health. Most of them show this pattern where they're decreased or changed in an adverse way with sedentary aging, but preserved with exercise. So we think exercise is having an important role on mitochondrial health. So just a couple of points about summary, and then one little piece that I put in for Marcus and Gary at the very end here. So this sort of summarizes what we've talked about so far. Regular aerobic exercise exerts a very remarkable preventive effect in preventing the development of arterial dysfunction with aging. It also restores much, if not all, of function that is lost as a consequence of advancing age. And this probably explains a big part of the preventive aspects of physical activity, not only against cardiovascular disease, but all of these common disorders of advancing age. And we think how exercise is exerting these beneficial effects involves suppression of the inflammation that develops in your arteries with age. It also suppresses the development of superoxide-associated oxidative stress and the downstream influences on nitric oxide and changes to the extracellular matrix. Exercise also appears to exert a direct protective effect on aging arteries against a variety of adverse factors that would do our arteries harm. And exercise, we believe, exerts at least some of these beneficial effects by preventing arterial mitochondrial dysfunction with advancing age and the resulting excessive superoxide production from those dysfunctional mitochondria. And what we're doing now, and where we're going a little bit with this work, involves the molecular transducers of exercise. And I use the term exercise instead of physical activity. No, it was already in there before the conversation with Marcus and Gary last night. But anyway, this is one direction that we're taking these data. And what we're using is high throughput molecular analyses to try to gain further insight into the mechanisms of these very protective effects of exercise. We have data on several of these, but exercise data right now, as I'm here today, on 2 of these omics. And let me just show you just one slide of data on our data on epigenetics. So we took blood samples from these 3 groups, young and older sedentary adults and older trained adults, and we isolated peripheral blood mononuclear cells. And we looked at the methylation marks from DNA extracted from those peripheral blood mononuclear cells. And we have methylation data on about 500,000 sites. And what we found when we did the analysis was 3 patterns or signatures of these epigenetic marks across these 3 groups. So here's pattern 1. It's very interesting. Pattern 1, we see an influence of age, but not exercise. So here's just a heat map of the epigenetic marks. These are subjects going from left to right, individual subjects. And this is just a sample, obviously. But you see that the 2 older groups have fairly similar epigenetic marks versus the young sedentary adults. So in this pattern, again, aging, but exercise doesn't appear to have much of an effect. This is the second pattern. We see no effect of aging, but we see an example of an effect of exercise because the data from the older trained look very different from the young and older sedentary groups. Most interestingly, a third pattern that has emerged is this one, where we see an aging effect, and then in the older trained adults, we see at least a partial prevention of this aging effect. So this heat map doesn't do it completely, fully justice, but most of those marks are preserved if we compare them to the young adult group. So this suggests a prevention of these epigenetic changes with aging, at least in these cell samples. And so about 15% of all the methylation marks that we assess follow this pattern. And the last thing I'll say, and this is interesting to us in the context of this discussion today, all of those epigenetic marks in that pattern are associated with changes in vascular dysfunction, changes in vascular function with aging, and the influence of aerobic exercise in preventing those changes. So these marks are somehow linked to, in particular, the endothelial dysfunction that we see with aging and the prevention of that dysfunction that we see when people exercise throughout their lifetime. And just want to thank our funding institutions and thank you for your patience, and happy to take any questions. Thanks.